So good afternoon, everybody. I'd like to welcome you to the 16th Annual Schwartz Mind-Brain Lecture. For those of you who were expecting Lorna Roll, who's the chairman of the department who organized this lecture, poor Lorna is flat on her back with a disc problem, a herniated disc, and unfortunately is not able to be here. However, she has substituted with somebody who's almost the same. I am L-O-R-N-E instead of L-O-R-N-A, so it's almost the same. Anyway, I'm very pleased to be up here to introduce Dr. Donahue, who is, as I said, the 16th Annual Schwartz Mind-Brain Lecturer. This lecture series is sponsored by Dr. Jerry Schwartz and the Schwartz Foundation, and it's hosted by the Department of Neurobiology and Behavior here at Stony Brook. It brings groundbreaking new work in the area of brain science to the attention of the Stony Brook University community at large. Now, the breadth of topics that are addressed and the outstanding research carried out by leading investigators invited to give these talks is our way of sharing some of the excitement that pervades the field of neuroscience today. This lecture will expand your horizons by demonstrating astonishing new approaches to coping with severe neurologic disease or injury. Dr. John Donahue will take us on a tour of remarkable new technologies that he and his associates have developed to literally turn thoughts recorded and decoded from electrical activity in the cortex into actions, such as controlling a computer or a computer-assisted robotic device. Dr. Donahue received his undergraduate degree from Boston University and his graduate training at Brown University. After postdoctoral training with Dr. Edward Evarts at the NIH and Dr. Stephen Kitai at Michigan State University, Dr. Donahue returned to Brown and rose quickly through the ranks to his current position as the Henry Merritt Riston Professor of Neuroscience. Dr. Donahue is a true pioneer in the field of neuroscience education and training. He was the founder and first chair of the highly regarded Department of Neuroscience at Brown University. He established the Brain Science Program at Brown, one of the first interdepartmental graduate programs in neuroscience. In 2010, 2 years ago, Dr. Donahue became the founding director of the Brown Institute for Brain Science. He's been the recipient of numerous awards. He's a fellow of the AAAS and the American Academy of Arts and Sciences. He has received the Roche Nature Medicine Translational Medicine Award. He's also received numerous awards for excellence in teaching, as well as special recognition awards from the U.S. Congress, Wired Magazine, and Discover Magazine for his innovative translational research in what might be called neurobotics. He has published well over 100 papers in top-notch journals. He has communicated advances to the field, to the public via such venues as 60 Minutes and Charlie Rose, and has been the subject of numerous New York Times interviews. It is my great pleasure to introduce Dr. John Donahue, the 16th Annual Schwartz Foundation Mind-Brain Lecturer.
Well, thank you everybody for coming, and I appreciate Lauren's kind introduction, and I want to thank Lauren for escorting me and taking care of me today, but also I want to thank Lorna, who tried very desperately to get here, but with her disc it was really better that she didn't come. And I also want to thank all the faculty and students and staff that helped make my visit today really wonderful, and really enjoyed visiting everybody here. So what I want to talk about is this idea that we're in the dawn of an age of neurotechnology, where we have devices coming that interface directly with the nervous system, some of which are things that will help diagnose and treat nervous system disorders. But one of the exciting things that I want to emphasize is those devices that will actually help us restore lost function, like blindness or loss of hearing or even loss of movement, which is the thing I'm going to talk most about. So the The history of this is kind of interesting, and it harkens back to another medical device, and I won't ask people to actually say the answer, but I'm just curious if does anybody recognize, if you think you really truly know what that object is, just raise your hand. A couple, but very few people. So that actually is a cardiac pacemaker from about the 1950s. And characteristic of a cardiac pacemaker in those days was that it consisted of wires that went into this gentleman's chest, into his heart, to pace him because he had an arrhythmia. And the electrodes, or the devices, came out, and they went to this sort of cart of electronics, and that cart of electronics contained a stimulator to electrically stimulate his heart that didn't beat properly, didn't pace properly, and an oscilloscope because the engineers and clinicians didn't know exactly what to do, so they kept everything on the outside so they could actually twiddle the dials and have the correct or appropriate kind of heartbeat rate for this individual. Now, as you know, in about 20 years, we transformed this kind of technology to something where all of this stuff was put inside the body in form of a cardiac pacemaker, which today is something that's routinely put in the body to pace the heart. So neurotechnology is a technology that some of it is actually way back here in the carts of electronics and not knowing exactly what we're doing, although as you'll see a little bit has moved towards the sort of advanced cardiac pacemaker. But the revolution in cardiac medicine is something that is now occurring in neurotechnology and neurology. And what's very special about this field is that it requires the interaction of fundamental neuroscience all the way bridging to clinical applications. And it is really something that is at the intersection of not only life sciences and clinical medicine, but also the physical sciences, which is really in the spirit of the Schwartz Foundation that's supporting this talk. And although it's a bit washed out here, this diagram is showing you how the fundamental neuroscience is interacting with engineers, mathematics, and computer scientists to get to clinical applications. And another point I'll make towards the end is that, in fact, what we're learning from these clinical applications of human devices is actually informing us about how the human brain is working. And again, as I said, I'll point that out. So what I'd like to do is to show you a few examples of the kinds of technologies that are developing, and specifically emphasize the work that I've been doing the past 10 years or so. So neural interfaces come in two basic varieties. Those that try to put information into the nervous system, usually by electrical stimulation, and those that take information out of the nervous system. So things that put information in are being used to restore lost sensory function, like hearing, and I'll show you an example of that, or for therapy, a kind of neuromodulation, which is effectively like trying to tickle the nervous system back into normal function using electrical pulses. Sensing or reading out the information is being used for evaluation. In fact, something like that's been around for a long time if you've ever had EEG or a sleep study where they measure brain electrical activity. And the other type of use of sensing is to restore movement, and I'll explain what that means in a bit. So I'll give you some examples of getting information into the nervous system. So one of these is the cochlear implant. It's been implanted now in more than 170,000 people, so this is really a mature technology. It is something in which a noodle-sized electrode is passed up into the cochlea, which is this snail-like organ inside your head that has hairs in it that pick up sound and transmit it into the brain. In many kinds of hearing loss, those hairs degenerate, they die. And so there's nerves there to conduct impulses into the nervous system, but in fact there's no sensors to pick up the sound. So what this electrical stimulation electrode does that's snaked up inside this cochlea is it has a small number of channels. We're actually replacing 40,000 channels with about a dozen channels, and it stimulates the nerve endings here, and impulses go into the nervous system, and these are impulses that are related to sound on the outside that are picked up by a microphone. Now, you would say, how could this possibly be so elegant to allow you to hear? But in fact, it works very well, although it takes some time to learn to interpret those electrical impulses as sound. As I said, about 170,000 people, including many children, are benefiting today by this technology, which is produced by at least 3 companies that manufacture and sell them. Another kind of stimulating technology that is at a much earlier stage is what's called a retinal prosthesis, or an artificial eye or vision, which takes the idea that we can make a miniaturized camera, transmit that to a little plate of electrodes inside the eye, and those tiny electrodes will electrically stimulate nerve endings that are connected to the central nervous system in an eye that has lost its receptors. Its light receptors have died by diseases like macular degeneration or retinitis pigmentosa. So those signals, when the light images are transformed into a pattern of stimulation, go into the brain, and people can see. Now, this is a very small number of people that have been tried in this kind of technology. I think there's something about like 60 people in the world that are in clinical trials. Here you can see the little plate of electrodes. And those people can't see like a flower bunch like we'd like them to see, but see sort of dots. Actually, this ceiling up here might be a good image for what people see. Little white spots against a black background, but people can see a white door against a black wall, or even sometimes crude letters. So this is an early technology, but it is promising to help blind people see again. Now, as I said, another kind of stimulation is neuromodulation. Probably the most successful device is one called deep brain stimulation for Parkinson's disease. Parkinson's disease, as most of you know, is a disease where there's a chemical in the brain, dopamine, that's lost. As a result, a person becomes rigid and has a tremor, and they're very slow to move. In this movie, you can see the lady walking in the shuffle gait, shaking, very slow and rigid. And it's a progressive degenerative disease, and thanks to the work of Benabid, it was discovered that you could pass stimulating electrodes, again about the size of a noodle, down into something called the subthalamic nucleus. It's a little tic-tac candy-sized structure in your brain, about 2.5 inches in, and if these electrodes are put in and stimulating currents are turned on through this tiny electrode, the tremor and rigidity go away, and you can see what happens when the stimulation is turned on. Here's this lady walking with the stimulation on. So this is a really remarkable technology. It has a big effect on at least some of the people who have Parkinson's disease. It doesn't wipe out the ability to, or requirement for medication, but it has a profound effect on their ability to function in everyday life. Now, most people are surprised to learn that, in fact, there's more than 80,000 people in the world walking around with deep brain stimulation. This is brain surgery. It's, as I said, these sort of 2 noodle-sized electrodes going 2.5 inches into the brain. But it's a really clear demonstration how you can modulate the brain back into a healthy state with stimulation. And there are many new applications coming for this. And so I'll give you a couple of examples, because I think this is such an exciting technology where you can put these electrodes into the brain and treat disorders. So another one is dystonia. It's a disorder of muscle tone where the muscles stay contracted. Here's a young woman who has dystonia. So you can see she's twisted up. She has a deep brain stimulator placed in a slightly different part of her brain, and you'll see in a couple of seconds. So she can't move out of that position. Her muscles stay like that. And after the stimulation, she's able to walk, and it's really treated her disorder rather successfully. Now, this doesn't work for everyone with dystonia, but again, it's one of these remarkable ways of neurotechnology transforming someone's life. So currently, there's work going on in using treatments for obsessive compulsive disorder, or OCD, where people have habits that they can't stop. For depression, there's a clinical trial going on to help people with very severe depression, and this has actually been shown to work at least in a few individuals. And there's even a plan of a trial in the coming year to treat dementia or memory loss with deep brain stimulation in another part of the brain. So these are all promising but very early stage. So these are all examples of putting information in. So now I want to spend the rest of the time talking about other neurotechnologies, or a neurotechnology, to read out signals, in particular the work that I've been doing to get control signals out of the brain to restore movement for people who are paralyzed, in this case severely paralyzed. So the best thing to do, I think, to start, I'll turn this sound off for a second just because it gets annoying after a bit. So this is to show you what a neural interface system is. In fact, our BrainGate neural interface system. This is a young man playing a video game, and his job is to hit these treasure chests and stay away from the goblins. It's called He-Man, according to one of my students who made this game. Now, his performance is okay for a video game, but this is Matt Nagel, our patient called S1. Matt Nagel was stabbed in the neck in the second cervical vertebra, high in his neck, and he's completely paralyzed. He's unable to move his arms and legs at all. And so he's playing this video game using a tiny chip we've implanted in his brain that's picking up the signals about his intentions to move his arm. And then that is coming outside, and we're translating that into a command signal that's going to a computer that thinks it's getting a mouse input, and that mouse input is then is controlling the cursor. So he's able to play the video game merely by thinking about moving his arm. So we're bypassing the bad inoperable circuitry in his nervous system, his inability to move his muscles, and going directly from brain to computer. So this technology is called a brain-computer interface, a neuromotor prosthesis, or a brain-machine interface. And we've named our version of this thing BrainGate. It's sort of easier to say. So what I'd like to do is tell you, this isn't magic, and it isn't. It has real solid science behind it, we believe, and so I'd like to explain how we could do this, hopefully to make you a more informed neuroscientist by the end of the lecture, and then also to show you where we might be going with this kind of technology. So just by way of introduction, I just want to remind everybody that paralysis is not a disease. It's the consequence of very many disorders like the ones listed here. But by and large, what happens in many forms of paralysis is the brain is actually functional, it's operable, and but what has happened is the pathways from the brain to the spinal cord, or the spinal cord to the muscles, have been damaged in some way. So they can be damaged, as in Matt. He had a knife injury that cut the pathway from the brain to the spinal cord. Another type of injury I'll talk about, called a brainstem stroke, is a stroke in the part of the brain that connects the cerebrum up here to the spinal cord, and it actually leaves people not only unable to move, but typically unable to speak, and in the worst case, the people are locked in. All they can do is move their eyes and nothing else. Another type of loss is the messages can't get from the spinal cord to the muscles. ALS, or Lou Gehrig's disease, is an example of that, where the neurons, the cells that connect the spinal cord to the muscles, die. And so therefore, even though the commands get to the spinal cord, they can't get out. And finally, we can even think of limb loss as a kind of paralysis, because you can create messages, I want to move, but in fact, the limb is no longer there to be moved. So there's an estimate, I think that there's about over 5 million people in the US that are afflicted by some form of paralysis. The numbers that have what's called tetraplegia, or some people know it as quadriplegia, the inability to move all arms and legs, is a smaller number, but it is an extraordinarily debilitating condition, because you become reliant on other people to do everything for you, including drink a glass of water, groom, or any other activities of daily life. So there are a number of laboratories around the world that are working on various aids to help individuals, various neurotechnologies. And so many of these BCI, brain-computer interface labs, are working on simple switches or other kinds of technology, like you can wiggle your eyebrow, if you can still do that, and that can click a switch and something can happen. But what we're trying to do with BrainGate is actually reconnect the part of the brain that actually controls the arm back to the outside world, so that you can do the same functions that you would ordinarily do with something else. And I'll show you examples of what those are. So our challenges with this, of recreating the arm and hand action, are to make something that's safe, that is as good as your arm and your hand, that's natural to use, that it doesn't really require all kinds of training and really high concentration, something that you can always use whenever you want, that it lasts a long time. But like in Matt's case, you wouldn't want to have brain surgery every few months if it required putting a new sensor in. You want a sensor to last. It should be portable so the person can walk around, and cosmetically acceptable, that is, it shouldn't look weird. And you'll see we haven't achieved all of these, although I will mention in the beginning, safety profile looks pretty good in what's an ongoing clinical trial. So if we could do that, there are a number of things we could do to help people who are paralyzed. So one would be to communicate. We could take the brain signal and connect it to a computer so they could type and communicate with others. We could connect that to an assistive robot, and there are really fancy assistive robots like the one you see in this video that could serve you a glass of water if you couldn't do it yourself. You could connect that signal in an amputee to a prosthetic limb, and there are prosthetic limbs that are coming that are really extraordinary, that can replicate many of the functions of the hand, but this gentleman isn't using his brain to control that arm. He's actually wiggling his toes and pressing on switches in his shoes to make those arms move. Or we could even try to reconnect the brain back to the muscles, and there are technologies already in place that could be implanted in muscles that could stimulate muscles, and imagine we could connect the brain back to the muscles and have the brain control them again, but now not with nerves, but with wires. So in order to do that, one of the things we would have to know is, what is the target? Where would you get these signals? And so I'm going to show you where in fact we've decided to pick up signals. The second thing we'll have to do is find some way of sensing this information. How can you get the correct information, the amount of information you need to actually do something like carry out arm and hand actions? And then finally, you're going to have to solve a problem which is we call decoding. That is, the brain's information stream that comes out, as you'll see, as a bunch of electrical impulses. We have to turn those into something that is in fact a meaningful signal that can be used to operate other devices. So we have to decode or translate those signals. And then once we have those, we can connect to all these devices. So I'm going to walk you through our thinking of how we wound up with identifying the target and the kind of sensor we need and how we decode. So we've known actually now for well over 100 years that the target of our target is the primary motor cortex arm area. So in all of us, there's a region of our brain that controls our arm. And it comes from a strip called the motor cortex that sort of starts up near the crown of your head and goes down towards your jawbone. It's about 1 inch wide. And it's divided topographically in a way that the third that is closest to the top of your head controls your leg, the next third controls your arm, and the most lateral third controls your face. So we know the arm area is right in here. So this is the territory where commands to move your hand, just like I'm moving my arm around, are coming from that region. The other thing is that we know a lot from animal research about what goes on in that area. So I'm not going to explain exactly all the details of what's going on in this picture, but just to explain to you that thanks to many years of animal research in which monkeys played video games, we know that in fact cells in the motor cortex actually start to do their thing, they're active, before you start to move, as if they're commanding it. So we know that they're commanding it by this activity. So this is some kind of neural activity that's starting before the monkey actually started to move, when he was making sort of a task where he would just point a cursor and move his arm over to point at a little target over here. In addition, we've also learned from many years of basic science that the information is not only just I want to move, but there's information about which way you want to move. And this little circle plot here is what's called the directional tuning of a neuron. And all it means is as that monkey moved to targets in different locations, like up and to the left here or down and to the right, the neuron discharged differently. It had different activity. So you can see up here by this little histogram, whatever it's doing, it didn't fire very much, but down over here it was much more active. So there's information about which way you want to move in the arm. So this information tells us that we could probably extract this information and use it to help patients who are still, who would like to move but can't move, and this would be the information in neurons that would be valuable to do that. So we know a lot. Now, what are the signals like? Well, we've learned a lot about neural signals, again, from many years of basic science research. And here are the two major kinds of signals that are present in your brain. So the brain uses electrical signals to communicate around the nervous system, and there are two basic types that come out. One is called a field potential, and the field potential is sort of this squiggly line here, and it represents, for those of you who are interested, current flow around of large numbers of neurons. And the other type of signal, which you can just by changing your filter settings see in the electrical record, is called a spike. There are these tick marks, these little events that occur, and these are impulses that are coming out of neurons, out of single cells. And the only reason I show you this video up here is to say whatever they're doing, they look different. That is, the spike is doing something here, and the field potential is doing something different. So there are at least two different kinds of signals. So I want you to appreciate that. Now, what we know is that the detailed information of how the brain operates is present in this pattern of spiking in neurons. What that means is that everything that goes on in your brain, whether it's seeing, hearing, thinking, or moving, is communicated by neurons that are using these impulses to communicate with each other. So we've focused on these action potentials, or spikes. They have two names, either spikes or action potentials, as the code of what's going on in the brain. And it's well known that there's detailed information in these spikes. The field potentials, which I won't talk about, are another sort of rich source of signals that we personally haven't exploited, but others are interested in. So one of the problems, if you want to get spikes as the source of information, is that they're not available outside your head. The only way to get them is to go inside. So the device that you need is something called a microelectrode. It's this hair-thin metal, almost a wire. It has to be introduced into the brain and nuzzled up close to a neuron. And when it's very close to a neuron, that is not more than a hair's width away, it can pick up these electrical impulses. And these impulses, as I said, pick up this signal. But this is really the message from one cell. And in fact, the problem is that we need to really pick up patterns of lots of cells. And just to give you a sense of what I mean by patterns, we know that there's information in patterns. This is the kind of pattern you start to see all over the place now, called a QR code. The QR code is a kind of two-dimensional barcode. Now, neurons are pretty much putting out patterns that are like this. So think of each little black or white square as one of those impulses coming out of a neuron. But it isn't just that it's a black or white pattern, but it's also evolving in time. So it's like QR codes that are playing in a movie. So that's the code that's coming out of the brain. That's the code that we have to decode or make sense out of in terms of what are the patterns and how do they relate to movement. And I'll explain to you, I hope, a simple way of how those patterns emerge. But a big problem is, how can you actually pick up lots of neurons at a time? I said to get one neuron, you have to put a little electrode in. We had to invent an array of electrodes that could pick up several, so we could look at this QR code-like pattern that's coming out. So starting with the original design by Dick Norman, and then later in my lab, and then ultimately produced by a company I started and then transferred over to another company called BlackRock, is this device called a microelectrode array. It looks rather big here, but it's actually the size of an M&M, about 4 by 4 millimeters. So this is 100 of those hair-thin electrodes. It's introduced into the brain. First, we tried it in monkeys, and then later it's now used in humans. And it consists of the array that's inserted into the outside of the brain, called the cerebral cortex. The signals are brought up by a cable that comes out to a connector. The connector goes through the skin, and this penny-sized connector has 100 connections on it, and that signal is brought out to the outside world. So with all of this technology in the late 1990s and the early 2000s, we said, well, we've got all this stuff, and we have this idea that we might be able to decode information. Could we, in fact, show that this is even feasible in an able-bodied monkey? So what we did was we implanted an electrode array in the arm area of the motor cortex, and while the monkey played a video game, in this case, the video game was, there was a red target that appeared on the screen. A green dot was reflection of the monkey's mouse cursor. He was holding onto this little arm, but it's basically like a computer mouse. And his job was to simply smash the green dot into the red dot. And when he did that, he got a drop of juice as a reward. But what we did was we said, okay, what's the pattern of activity that's going on in his brain when he's actually moving? If we can decode that, we can then pull the plug from the mouse into the computer and just substitute the brain signal directly in. And in fact, that's what we did. And that's what actually Misha Soroya did in this 2002 study. And by going directly in, the monkey was now using the output of his brain to drive this cursor. And actually what you've been looking at all along is the monkey playing the video game with his brain and not with his arm. That green dot is under brain control. So at that point, we knew the sensor worked, we knew where to go in the brain, and we knew that we could, at least in principle, in an able-bodied monkey, be able to show that you could use that activity to perform a task. Well, that would be very helpful for a paralyzed person. So we then said, okay, let's devise a system, and we put together a system that consisted of the same electrode array coming out through the plug on the head. It has a pattern of electrical activity that's taken out. It's decoded through a cart full of electronics, and what comes out is a cursor on a screen that the person can control a computer simply by thinking about moving their arm, even though they can't really do that. And so I hope, if you've been getting my connection here, cart of electronics, hole through the skin, it really looks like the old-fashioned cardiac pacemaker. So that's where we are with this kind of technology. And So with FDA approval, what we've done is we've implanted so far 7 of 15 patients were allowed to enroll in this study. The first one was done in 2004. That's Matt Nagel. He was one of 2 patients with spinal cord injury. They're tetraplegic, but he can speak, and you'll hear him speak. And the other patient that I'm going to talk about a bit, and you'll see her in a couple of the videos, is patient S3. Her name is Kathy. She's appeared on 60 Minutes. She had a brainstem stroke, so she can move her head, but she can't speak. But she can smile and at us and very nicely. And she can't move her arms and legs either. We've also studied 3 patients with ALS, and I'll just mention that very briefly when we go by. Now, I know it's before dinner, but I'm going to show you brain surgery. I hope that's okay. So the idea is, can we do this in humans? So in fact, a nice anatomical event in humans is that there's this little bump here called the knob, and guess what? The knob is the arm area of the motor cortex in humans. It's been well documented in a variety of studies that that's the right place to go. So we knew exactly where to go based on an image. This is one image. The surgeon makes a small hole in the brain, the skull, opens this membrane called the dura, and you'll see that the electrode array is just simply tapped into the brain. And I'll play that video. You can see it. It's almost a non-event. It'll just be a quick tap, which happens there. When that's all over with, it's inserted, and you can see that it just sits on top of the brain. The surgeon closes everything up, and the only thing that's left is the penny-sized connector coming through the skin, but everything is closed. So that's how the patients are left. And we are able then to go to their homes and set up the recordings and record from their brain and see what happens. Now, when we first did this, we were very concerned because in the case of in all of our patients, they were at least 2 years after they had had their injury, whatever it was. So they hadn't moved at all in 2 years. And in Kathy's case, the brainstem stroke patient, it had been 14 years since she had done anything. So we were worried that this part of the brain might just shut down and have no activity at all. But even if you don't know what this is, you can presume that this is brain activity. This is in our ALS patient. And what you're going to see, and I'll show you this a couple of times, this pink check mark is one of those spikes, one of those impulses out of a cell, and it's spread out in time so it has a shape. So this is actually 1,000th of a second or so long here. It's playing here. You can see the spikes going by, and you can see them going here. You don't have to worry about all this. It's just that there's spiking activity. But this little set of black boxes with little check marks in them is actually each electrode's recording, and all I want you to see is there's a little colored check mark in each box. There's lots of cells. So the first big important finding is that people who have long-standing paralysis still have activity in their brain. But even what's more important is that this activity actually does something. That is, can a person who's been paralyzed for a long time simply think about moving and have their brain do something? So here what I'm going to do is I'm going to let you hear a technician talking to Kathy while you look at, here's this action potential, and really all you need to do is listen in. And he's going to tell her to imagine closing her hand, relaxing, or opening her hand. And what you're going to hear is the popping of the cell, the activity of the cell, change its activity. It's going to go from silent when he says close to very active when he says open, and in between when he says relax. So you're actually listening into her thoughts of movement. Now this is one brain cell that you're listening in on. So
relax. Now imagine you're opening your hand. Relax. Close your hand. Relax. Open your hand.
So I think you can hear that change in clicking was immediately the sound to us that even in a brain that had long-standing paralysis, it was completely possible to get information about what was intended. Here, Kathy imagining opening and closing her hand. So this meant that, in fact, there was viable activity surviving. So one of the issues you might ask is, well, how could you possibly get from there to what you want to do is recreate arm action? So we had to think of a way. So what we did was we would play a video to our patients, and the video would show a cursor moving out to these targets back and forth, and we would say, you imagine that you're actually making these actions. And while they did that, we recorded from their motor cortex. So if the motor cortex was engaged, what we would see is that these cells would be active. And in fact, here's an example of a cell from Kathy's brain in which the cell was, in fact, and as many of them are, directionally tuned. Just like I showed you in the monkey, the cell fires more when she's imagining moving the cursor over here than when she's moving the cursor over there. So in fact, all we have to do is build a mapping that says, how are these cells coding for direction, and how does that code relate to which way you want to move the cursor? So for those who are interested in the mathematics, I'll flash by this quickly, but I took this quote from the Schwartz Foundation webpage about this talk, and said much of what the brain does could be modeled computationally. So we depend on an accurate computational model of what all the cells are doing and how their firing relates to what the person wants to do. And I'll just tell you, there's lots of work going on, and I won't go into it. What I'll do is I'll try to give you an intuition for how this works. So now imagine you could record from just one cell in my brain, and I was moving my hand to the left and then moving my hand to the right. And every time I move my hand to the left, the cell, or I imagine moving along with this cursor, the cell would fire 5 spikes. And every time I move my hand to the right, it would fire 2 spikes. So now you have a code. 5 means left, 2 means right. So that's effectively what we do. We just count up, the computer actually counts up in these really short time intervals how much the count of spikes, and says, okay, if you get 5, it means left. And now increment the cursor, when the person's controlling it, a small bit to the left. Now, in fact, neurons are not at all that reliable, that on different trials the number of spikes will vary, but by averaging across a lot of neurons, we can actually get a pretty good estimate of the code of left, right, up, down, or motion of a cursor across a screen. So with that, we're able to feed it into a computer, as I showed you earlier, and have the computer think it's actually getting commands to move a cursor. So the person thinks they're moving their arm, we decode it, and they move a cursor. So I'm going to let Matt show you, some of you have seen this on the internet, it's an old movie now from 2006, and he's going to do 2 things. One, he's going to use a cursor to open an email message, and he simply has to pass the cursor over the email message. And then the other thing I'm going to have him do is open and draw circles, and you're going to see him potentially learning something new as he tries to draw the circle.
Desktop. What are you going to do first?
I'm going to open my email first.
Okay.
So this is the email. You can't see
First
too well.
one.
You're going to open the first one.
And it says, congratulations, you are doing a great job.
Very good.
I'm going to open the second email.
Okay.
This one says, hi, we'll talk soon.
Great. Now can you exit back to the cybernetics
I'm
desktop?
going to exit.
You're going to exit. Excellent. So what's next?
Next, I'd like to draw a circle.
You're going to draw a circle.
This is a paint program here.
I'm going to open a page.
So it's an inkwell and an eraser. And he'll try 3 times. So
Let me try that again. I erased it.
All right, let's try it again.
this is the first time he ever tried anything like this, too.
Whoops. You avoided the eraser that time.
That's the best circle I can do. I'm going to exit.
Okay.
I'm going to draw a circle.
All right. Excellent. Now let's try going back to the desktop.
So he was able to do some very basic functions, but as you can see, the cursor was a little bit wobbly and not perfect. So, but given that it's only a small number of neurons, brain cells, that are trying to operate a cursor, usually when you control your arm, there are millions of cells engaged. So we're actually doing an amazingly good job of recreating a desired movement of the hand onto a computer. So when we did these early studies, we were very excited and said, what else can you control? And so we came up with this mechanical hand, and we asked Matt, this was the first time he saw it, it's sitting here on his lap on a blanket, he's up here, and we just asked him, we said, imagine moving a cursor up and down, and we'll connect it up to the arm so you can actually see something move as a consequence of your thinking. And actually, it's not a very impressive demonstration of anything, but if you listen to Matt's response to what happens when, for the first time in 3 years here, I think, he actually moves something. It was rather a big event for him.
Close. Holy shit. Close. Nice. Open. Close. Not bad, man. Not bad at all.
So you saw, if you go on the web, that movie is on the web, but it's edited. So you got the uncensored version. So, as I said, the cursor control, and this was true for all our patients, interestingly for ALS, spinal cord injury, or stroke, they could all control the cursor, and originally it was all very wobbly, but thanks to building better decoders and things I won't have time to go into, working especially with Michael Black and his collaborators, we've come up with much better decoders. And so here's a more updated version where you can see the movement of the cursor is smoother, and in fact we're capitalizing on the fact that we can capture both where I want to move my hand and I want to squeeze my hand as a click signal. So this is Kathy doing point and click actions just like you do with your computer mouse on a screen. So she can go to a target, stop on it, click indicating that's the one she wants to select, and then go back. You can see it's, again, not perfect and not as fast as you or me, but much better control. And this is thanks to better decoding algorithms, better computer code and better mathematical algorithms that allow us to decode those signals in a better way. Now, so with those, in the past several years, we've been working on ways of connecting these brain signals up to various kinds of devices that we would use to control a typing interface or various kinds of physical devices that emulate the control of an arm, and I'll talk about those in a minute. So I want to just show you one communication device that Dan Baescher and Lee Hochberg in the lab have developed. One is if you can point and click, you can type on a keyboard. So here's a keyboard. Kathy is typing the quick brown fox is lazy now. The way she does this is she has to go and select a key, click on it, and a bunch of words appear up here, and those words are like on your smartphone, they're predictors, and so she just has to go up, and she likes to select the word, because I notice when she's typing lazy, she goes right over the Y, but she heads up to the word lazy and clicks it. Now, one of the problems with this kind of keyboard is if you're just doing sort of one-fingered typing or one piece of your brain typing, you have to go back and forth because the keys typically alternate because of the way a keyboard is designed. So they developed a radio keyboard which is much more efficient. So she's now using this radio keyboard, so it's around in a circle. It's much like the text that's used on some of the earlier smartphones, and so she selects a letter, and then if she wants, she can say the word I want is in this list, and she can select that and the word appears, so she typed the word the. So she's much faster at typing, but again, not as fast as you or me. But this is a device we're doing to help people who, especially like Kathy, who can't communicate at all, to be able to communicate. So now, a bigger question, a cursor on a screen is one thing, but could you actually control something that's like an artificial arm, like a robot or a prosthetic limb, or even your own arm? And that actually challenged us in a way to say, this is really a big deal to try and get that much information of moving your arm around in space and grabbing something. That actually requires about 25 joints in your arm and is what's a 10-dimensional movement, moving around your hand in space and opening and closing your hand accurately. So we went back to the monkey preparation, and we asked the question, well, if we had monkeys trying to reach out and catch toys in space, is there enough information if we record from their arm area motor cortex to actually reconstruct all of that? Is there information about the whole reach and the whole grasp, or is it just limited to very simple commands? So what Carlos Vargas Irwin did is he trained a monkey to reach out and grab little toys swinging on a string. So here's the toy, and this is a video capture image of what the monkey was actually doing, and we're able to capture that from our motion capture. So this is an able-bodied monkey moving his arms around, capturing these objects. And at the same time, we recorded from the brain and again asked how much information is in the brain about everything that's going on there. And I will just, to summarize all that data, what we found is that from one little patch in the monkey's arm area motor cortex, we could reconstruct everything. So what do I mean? So here, what you can see, the ghosted image is what the monkey's arm was actually doing, and the more shaky solid image is what we were predicting from the brain activity. So if we were perfect, they would be completely superimposed. We're not perfect, but what you can see is, if you watch this, is that we're able to capture everything about where the arm is going in space, and even what the hand is doing, reaching and grasping. So this said to us, our little patch in a human motor cortex should have the same kind of information if the paralyzed person's brain is operating the same way as an able-bodied monkey. So recently we've been testing that idea by asking our participants to try to control a robotic arm. So this is the DLR robotic arm. It's just a commercially available arm with a specialized hand. It's produced by DLR, is the German space agency, and Patrick von der Smart is chief roboticist there, who helps us set this up. Here's Kathy sitting in her chair. She's been connected up with the technology that's coming off. Here's our cart of electronics. But we set it up, and our goal then was to see what could Kathy actually do. So we had her watch the arm and imagine she was controlling it. We built a decoder and asked her to control the arm. So I'm going to let you see her control. So, whoops, so the first thing, we just wanted to see, could she control the hand? So we had her imagine squeezing her own hand. And you'll see first in the video, I'm going to ask her, imagine closing your hand 3 times, and you'll see if the hand responds appropriately. And then I'll ask her to, then she'll be asked to move the arm.
Open and close the hand 3 times.
So that's Kathy imagining here a squeeze, and you can see she can open and close the hand. There's Patrick over there watching intently.
Can you try going down to the table again if you can?
So here's Katie, our technician, said, just try and lower this down. We had just shown her up and down movement, so this is only allowed to go up and down.
Okay, and now can you lift it up halfway? Can you lift it up all the way?
So with that, we saw that she was able to think and control this very complex robotic arm, which we were taking brain signals out, and it was going through something like a shoulder and an elbow and a wrist and this hand, and she was able to raise and lower the arm. But could she do something a little more challenging? So we asked her to pick up a wine glass on a table, move it around, and then put it back down again. And so here she is picking up this wine glass and moving it around. And so here's the wine glass. Let me start the movie. She's picked it up. And I have to say, we asked her first to put it down on the black circle, and she didn't. She decided to put it down on the blue circle. And you'll see Patrick walking over to get the glass because it's kind of near the edge, but Kathy told us later that she decided, well, that wasn't a very good job. So she went back over and adjusted it so it's a little bit better placed. So not only is she moving the glass, but she's actually is pretty good at placing it. So the promise here is really a lot of what she could possibly do to be able to interact with the world on her own. And we're actually pursuing that now, is asking how many kinds of useful activities, like being able to feed herself or groom herself, can you do with these robotic arms? And that's an area of active intensive investigation. Now, another concern is how long will these kinds of things last? We put an array in the brain. Will the brain actually keep this thing intact and allow it to work, especially since we're relying on very small electrical signals from the brain? So in terms of our study, a very important piece of it is how safe is it to have even this M&M size electrode array in your brain. And so we don't know the answer to that because we haven't finished our study, but in more than 10 years cumulatively across our patients, we've had no significant adverse events. It means that nothing bad has happened that we didn't think that wouldn't happen with any other person during their life that has these serious disorders. So the safety actually looks quite promising, but again, it needs to be looked at over the long term and in more patients. The other important part is, does it work over a long period of time? And there are big concerns. You know, is there tissue reaction? Do the cells die? Is there a scarring? Are there mechanical problems, like the electrode gets pulled out of the brain? Are there technical problems, which means that the materials degrade because they're inside the body? Or do brain cells change? Nobody's actually looked at human brains over many years and whether their cells actually change at the neuronal level. So all of these things are concerns. We're looking at all of them. Interestingly, we think this tissue reaction, at least of loss of cells and scarring around the electrode array, is not a big problem. But all of these other things have cropped up, and we're working on them, and I'll even mention some of the little challenges we've had. But the most important thing is that these electrode arrays remain functional for a long period of time. So Kathy has had her electrode array in for a long time. So here you can see her playing the video game, the same video game you know. And what we did is we actually did one study in which we said, okay, at 1,000 days, around 1,000 days, we're going to look at how well she can get to targets. And this is just the data from that study. And what you can see is in these, across day 999 to 1,003, she got from 83.7 targets to 100% of the targets. So she did pretty well. She got most of the targets. And the movie you're looking at, she was actually playing the game, and I think I'll start it up again here, is that this movie was taken at more than 5 years. So despite the fact that there is certainly some kinds of tissue reactions, some kinds of issues with these arrays, even at 5 years, this array is still working. So again, it's one patient. We don't have much other data, but so far it's at least promising that we can put an array in the brain and it can last for very long periods of time. Now, I want to make sure that you understand that not everything works well all the time. These little trails here are tracks made by the cursor that Kathy did on one day. And during the session, all of a sudden, she couldn't control very well anymore. You can see the cluster of traces up here means that she had some kind of bad control, and the cursor was drifting up to the corner. And if we look at the neuron, even though you don't know what's going on, you can see that these are all the spikes moving to different targets and when it worked, and these are all the spikes when it didn't, and all you have to see is there's something different looking over here than here. So something changed, and we're actually now looking into what is it that when occasionally we see these kinds of changes, what's actually going on. It could be that there's a technical problem that we can simply adjust, or it could be that the neurons are changing, and we have evidence that there's a little bit of both of these things kind of going on, and we still need to fix that. Now, the other thing is, if we're going to make this something that's practical in the long term, as I said at the beginning, we're going to need to get rid of the plug on the head. It doesn't look cosmetically nice, and it's kind of hard to have a technician set it up every day. We have to get rid of the technician in the room who can't always be there, and certainly get rid of all this clunky cart of electronics. So we're working towards wireless, automated, and miniaturized technology. And so here's sort of a vision of what we would like to do. Here's an electrode array attached to a paddle of electronics. And what the electronics would be is a plate that is in fact containing all the amplifier systems, and it could be, when the array is implanted in the brain, this plate of electronics would be mounted above the skull and wirelessly transmit through the skin. And so then you could close everything up and you would see nothing in the patients, and they could use it all the time because it would be transmitting. So thanks to an amazing group of people led by Arto Nurmiko, who's actually a photonics physicist at Brown, an engineer, we have developed a prototype that is now in testing in animals but not in people. And here it is. I think you can barely see. There's the array, the cable, and this little package of electronics, which for those of you who remember Zippo lighters, is about the size of a Zippo lighter. It's about the closest thing I can remember to that's a common everyday device that this is scaled with. And this device contains within it the electronics that are about the size of a DVD player, shrunken down to something the size of your thumbnail. It will pick up all the signals and send them to the outside world under battery power, and it all can be hidden under the skin. So this, as I said, is in animal testing, and it's not quite ready for people, but we're working on that, and these are just some of those waveforms recorded wirelessly through that system. So it's coming, but not there yet. So overall, what I've shown you that I think are big advances in the technology and also in our understanding of how the brain operates after damage or disease, is that the brain is still working, particularly the motor cortex is still working in a range of disorders, including stroke and ALS and spinal cord injury. And for those of you who actually understand these pathways, the lesions, the stroke or the spinal cord injury, have actually cut the outputs of the axons of these cells. So these cells, many of them have been directly damaged, yet they still seem to function, which is a surprise for us. The other thing that was a critical advance is that intention works. If you merely think about moving in the part of the brain that's supposed to actually control movement, it still works even though you're paralyzed. That it is actually possible to decode. We can create algorithms thanks to our applied math and computer science friends. We can actually take that complex QR pattern of activity that's emerging out of the brain and translate it into something that can be a point and a click or a reach and a grasp. It seems to be ready to use. That means that you don't have to learn anything. We just have to build this initial mapping, and then you can use it right away, although we do think that learning will help, but we haven't studied it. It has to be long-lasting. We have a little bit of evidence, 5 years in one patient, but we really need to know whether this will really last for years. As I said, it's useful. People can use it for communication or for reaching and grasping, like I said, with this robotic arm. But again, there are many challenges that remain, like making it wireless and automated and faster, so that it's not as slow and inaccurate as it is. And again, I emphasize that this technology is really still in the early phases. But it has an important benefit for people who are severely paralyzed. So one way to sort of get a sense of where we are in this is to compare it to science fiction. So I thought this is from a very famous Star Trek episode. This is Captain Pike, who's been completely paralyzed due to an injury, and he's in Stardate 3012.4's version of a brain-computer interface technology to communicate. So I'll let you see what he was able to do. A great melodrama.
You remember these gentlemen. They wanted to visit you. 2 flashes mean no.
So
I thought you might make an exception for them.
So for 1,000 years from now or so, that's pretty crummy technology, actually, I think. So then the other one is, of course, Star Wars. Those of you know the storyline here. Darth Vader, his father cuts his hand off, but he goes into the shop, and they literally put a new hand on that not only can move as dexterously as we can, but in fact, it actually feels just the same way we do, which is something we haven't accomplished at all. So this is maybe beyond where we are. So we're sort of someplace in between. We certainly don't have the Star Trek kind of simply 2 blinks mean yes and no. We have Kathy reaching and grasping for a glass on a table, but we haven't reached the sort of Star Wars version of this thing where people can get immediate replacements and do everything that your hand can do. So our long-term vision, or one of the important long-term visions, is that someday we will in fact be able to connect the brain up to a physical wired nervous system. As I said, this kind of wired nervous system does exist, but without a brain connection, so that someday, and this is, again, very far in the future, very speculative, but one day a person who is completely paralyzed would be unrecognizable from anyone else walking down the street, or this movie was showing someone playing basketball, that there'd be a complete restoration. Now this, as I said, is very fanciful, very far-fetched, because the amount of information that would be required, the amount of connections we believe is far beyond what we know now. What we'd like to do is to just get simply to help people who are paralyzed be able to carry out everyday functions, like being able to have a drink of water, or be able to groom themselves, or be able to type on a computer so they could go back to productive work. So what I've told you about is all the applications, emerging applications of neural interface systems. The stimulating, I told you that there's many applications of electrical stimulation or putting information inside the nervous system, like neuromodulation for Parkinson's disease and for hearing. There's also other applications for pain therapy, spinal cord regeneration, and there are many people receiving this kind of technology now, and many forms of this stimulation in development. On the other hand, sensing is much earlier, more like, as I said, the early cardiac pacemaker. I just told you about how we're using it for various forms of movement impairment. There are other applications where I think this technology will be expanded. So I'll give you just one example to end with, and that is in epilepsy. Imagine with with this kind of tiny sensor where you could pick up detailed activity of the brain. You could pick up the activity in a person who has epilepsy. And from this spiking activity, you could see that something's going wrong. And we know in epilepsy, the spiking activity, these action potentials, go from being very random in the way they fire to very periodic. They burst in repeated bursts. So imagine if you could have a detector inside that would warn a person that they were going to have a seizure in 15 or 20 minutes. Or you could tell them, you know, don't do anything dangerous when a seizure is about to come. Or better yet, you could tell them to take their medication, or even better than that, you could have a drug pump associated with this device, and it would deliver drugs to the seizure focus, and the person would never know they were about to have a seizure. And I'll tell you, as amazing it is now possible, but not practical yet, to mount a drug pump on the side of one of those electrode hairs at that scale. So it still impresses me that that could be done. It can't be done in an implantable device for humans yet, but it's getting there. So let me just show you one last slide, this recent data that just came out. And this, in fact, is from a human that had this array implanted for a short time for another reason. And it's a person who has epilepsy. Here is where they had a seizure. And what I want you to see is that this is 40 minutes before they had the seizure. And in fact, even though you don't really know what this record is, it's spiking, you can see something's going on. So it's very promising that with the same kind of technology and our knowledge of what's going on in the disease, we might be able to predict and inform a person about that they're about to have a seizure. And the early evidence is very tantalizing that these kinds of devices could be made. So I come here really as a representative of a multidisciplinary team that covers a broad range of activities. And so I'm, as I said, a representative, and I want to just acknowledge in the end here all of the people who have really contributed. So the leaders of the groups are up here. Arto Nurmiko, I mentioned, is really developing the implantable devices. Michael Black and Wilson Trucolo are developing the computational algorithms. Carlos, who was my postdoc and who's working in the lab, is now, he's a kind of Zen-ish kind of karate guy, and he's standing here between two pillars meditating in some way, the only picture I could find of him. And then I also want to mention Lee Hochberg, who is a physician scientist who works with me, who runs the clinical trial and is really a key partner in maintaining and running and driving this clinical trial. And then the group of people who I don't have time to name, who are our collaborators and students, who are really an extraordinary group of people that make this all possible. So with that, I'll say thank you very much. So I guess
Thank you very much, John. So we're going to have time for a few questions, but what I wanted to also invite you to is a reception that will be held upstairs on the upper level as soon as the questions are done. So those of you who'd like to meet John, please come by and say hello. So I'm just going to let John run the questions and for a few minutes. I don't know, are there microphones on the sides? Well, no one has said anything, so...
All right, I'll try to hear them and then repeat them if you can. That's a little. Yes, here.
It's really mind-boggling what may happen in the future, because at a certain point, these technologies may go into making people superhuman. So they could do something that are not accessible to a regular person today. So I imagine that at a certain point there may be even legal questions or ethical questions. Do you ever, think of these things?
So it's really, are there ethical implications for this technology? And certainly there are ethical implications, and we think about them a lot. And we teach our students that we are working on devices to help people who have disabilities recover independence and a free life. But in the future, there may be other applications which may augment human capabilities. So I think this is similar to the kinds of things we've seen with drugs, where drugs are given to people to augment them and they're used in other ways. So what it needs is a careful monitoring public discussion like this, so the public awareness of what's going on, and I think careful oversight by the scientific and responsible oversight by the scientific community, just like was done for recombinant DNA usage, to make sure that these are applied. But we will be faced, I'm certain, with ethical challenges on how these devices can be used and how they can be, if it's simply for patients to restore them to a regular function, able-bodied function, that's an easier one. But as soon as people try to apply them to augmentation, it gets more challenging. Of course, it requires brain surgery, so it's not an option. It's not something you can do at home. At least not for most of us, I think. Another question?
One over here, John.
Oh, yes.
In all the examples you showed us, the patient's feedback was visual. They were looking at what they were doing. Now, I can tie my shoes with my eyes closed. Do you have any examples where the feedback is something other than the visual, mechanical, or actually from the organ that's doing the motion itself?
No. So the question is, do we have any feedback? And the answer is no, other than vision. So that's how they assess their performance. And we do know that vision is very useful to them, and that's what allows them to control the cursor. So the obvious one would be to have them close their eyes and try to control the cursor with their eyes closed. And we haven't ever done that, surprisingly. So that's something that's actually underway. What we would also like to do is that, at least in the stroke patients, they actually do have sensory feedback. They can feel their limbs. So we could provide some form of tactile feedback. There are some animal experiments that have been begun to say that you can get information into the brain if you wanted to stimulate, or in the case of an intact person, they can actually feel. And this should be valuable and help them do a better job moving, but we haven't done it. In the back up there.
One of the amazing features of what you told us is that you can take 100 electrodes and put them into the brain, and apparently without exactly knowing where the electrodes are with respect to individual neurons, you can somehow manage to get a stable signal out of more or less 100 electrodes. And are able to keep this configuration tight so that you can reproducible get a system. Could you comment on that?
Sure. So the issue is stability. So if we used yesterday's model on tomorrow's session, it won't work as well. So we, in fact, build a new decoder every day. And the reason is that I mentioned that in order for the sensor to pick up the neuron's impulses, it has to be less than a hair's width away. So overnight, the brain's motion, the patient's motion will almost always move it. And so we just assume that it's moved enough that we have a different set of neurons. Now, it's actually being investigated, and a couple of labs have reported that something like 30% of the neurons stay around and the rest of them change. So we don't actually know in great detail how stable that signal is. So one of the other big challenges is to build what's called an adaptive decoder. So we have the computer that eventually would be inside the person, overseeing what's going on and changing things when it says, aha, we lost that cell, but we picked up a new one, we have to rebuild the model. The same thing is true of cardiac pacemakers now. I don't know, in cardiac defibrillators, it's monitoring the state of the heart, and in cases where it decides that your heart is no longer beating, it decides to defibrillate you. So we've seen that technology in something complex but simpler in cardiac technology, but not, it will need to come in our technology as well, for sure. I think up here, yes.
So going by your analogy, the Star Trek versus Star Wars theory on how you give the output versus both ways, the tactile input and output, do you think it might be necessary to investigate a two-way pathway instead of simply investigate, let's say, EEGs and your BrainGate technology, and instead integrate them at all times?
Sure. So I think it's a great question. The question is, you have two kinds of signals. You have the field potential, which might be EEG or ECOG or LFP. They have different names, but they're all that field potential signal I showed you, and spikes. And in fact, yes, that's exactly what we're doing. So we spent a long time, given that the dogma in the field is the code is spikes. So we first said, what can you get from that signal? But you'd be a fool to pass over another signal which has another band of information. So in fact, that's what we're doing. We're actually building what we call hybrid decoders, which take all the information we can find anywhere. We'll take anything. We're very opportunistic. And try to help, control things. So that's exactly what we're doing. And there are actually a couple of papers out in monkeys that we've published on the same topic. And up there.
A lot of your user interfaces, you said the patients over time, the control declines. Do you have any way of making the patients be able to calibrate the system so that they can better usable?
So the control doesn't really decline over time. Actually, the biggest problem we've seen so far is, in some sense it does. The 5-year-old array that was in Kathy, the materials had degraded, and that actually, some of the signals were lost. And we're investigating what it is about the materials. We have some sense. But actually, it's pretty hard. The body's a very harsh environment. And so it degrades the materials. And that leads to a degradation in the quality of the signal, which we clearly saw. So the important point was that it was good enough, even at 5 years, to create something where she couldn't do point-to-point movements. And even the robot arm control she did was 5 years after the implant. So it still works, but it isn't good enough yet. So one of our big challenge for sort of 10, 20-year lifetimes is to make materials that last, we think, rather than worrying about actually the brain's response to this. Yes?
You showed a connection between the BCI and robotic assist devices that were moving arms and whatnot. Do you have any plans in the works to necessarily incorporate skeletal robotic assist devices which can restore movement to people who are paralysis and off to walk again, hooking this BCI, this BrainGate, into something like that so that you could actuate an external skeleton and restore movement?
So the question is, could you use this device to control an exoskeleton that is sort of braces on the outside of the body? And so, as I said, our focus has been on the arm. And one of the reasons why we're focused on the arm is because walking is really complicated. And if you ask people who are paraplegic in a wheelchair, they get around pretty well in a wheelchair. Obviously, we'd like to restore locomotion. But our understanding of solving the problem of locomotion, movement, and then added to that balance, because it requires upright balance. So those are big challenges. Interestingly, in terms of how the brain controls those, in the cortex, there's not a lot of knowledge. At lower levels, we understand something. Those are harder targets. So then the second part of that is, what about an exoskeleton for the arm? And so it's a complicated question. There may be people who would like to have exoskeletons, but a big brace, a metal brace on the outside of the arm. I was privileged to talk to Christopher Reeve. He didn't want contraptions on the outside of his body. If he could have a device on a tabletop doing things for him. But what everybody really wants is, where it's possible, is to reanimate their own muscles. And that's this technique of connecting to what's called an FES system inside the arm, and therefore they move just like you and me. And as I said, our fanciful goal would be, you can't tell one person from another. Maybe their arm would move just as well as ours in 20, 30, 40 years. And then maybe after that, or maybe in parallel, people will develop walking and they'll be able to walk. But I'm very humbled by how big a problem it is to recreate walking from a person who has a spinal cord transection. Lauren.
So,
speaking of spinal cord transections, one of the leaders.
So what about handedness? You put this into the dominant hand, and if you do that, or if you put it into one side, and you show it a hand that looks like the same side hand, does it have more difficulty controlling that than the normally crossed hand that it would activate?
Yeah, so the question is, what about handedness, I think is the question. So we have, in our FDA trial, we had to make a very compelling argument why it was appropriate to actually put these into humans at all. So we had to decide, maybe 2 arrays would be better than 1, but if you don't know what it's like to stick one thing in, you don't want to stick 2 of them in. So we stuck with 1. Then we had to make the compelling argument, as I hope I did here, that there's a place you want to go and put it in. And you'd say, if you want to get the best possible function, I don't really know the answer, let's go with the dominant hemisphere. So we did that. So we have never, and we don't have any latitude to change that without a long interaction with the FDA, which I believe is appropriate, so we could go to the other side. So we've never done comparisons like that. We have the only thing I can tell you is that Matthew was left-handed. We went into his right hemisphere because it's crossed, and his control was ballpark the same. I think the noise in the signal makes it almost impossible for us to say anything meaningful about left versus right hemisphere control. I think we just had a flourish of other questions here.
You mentioned that some of these experiments don't work as well in some people as in others. Do you know why?
No, I wouldn't say, I'd say each day, some days work better than others. I would say, so the control has been about the same in all of our patients, which is very surprising. In ALS, not in a very subjective sense, we think the control has not been as good. And that, ALS is a degenerative disease, and I didn't mention, but in most people at some stage in the disease, the motor cortex begins to degenerate as well. So it may be degenerating there, and we don't actually know how much that's occurring. In fact, we were surprised at how much activity there appeared to be. So that's a question I can't answer right now. So. Sure, just the memory loss idea. So the idea is these are all devices to electrically stimulate, and there are a number of groups interested in what diseases or disorders can you treat by electrically stimulating some pathway. So in fact, the trial, as I know it, and I'm sort of conveying a secondhand story, but as I understand from Dr. Lozano, Anthony Lozano at Toronto, who did this work, there was a patient with morbid obesity. They got special permission to try stimulating in the hypothalamus and the feeding centers to suppress that person's appetite to help control their weight. They put the electrode in, they stimulated, and for about 2 weeks there was a suppression of appetite, but after that it went away. They did, as part of the normal testing for safety, they did a cognitive battery on the person to make sure they hadn't affected his memory. They studied that person before and afterwards, and in that individual they had a significant cognitive improvement. They got better. So then they realized that they were actually stimulating a pathway called the fornix that leads to the hippocampus, which is a memory center, and that led to the idea, that's why I said it's in planning, that perhaps we could treat memory decline or memory disorders by stimulating in that site. There's really, as far as I know, not much knowledge, but other people know more about this than I do. Other people have stimulated that pathway in animals, so if you know more, I'd be interested to talk to you afterwards. But the idea is that shocking that pathway would actually enhance memory, and there's an anecdotal case where something like that appeared to happen.
So I think we should move upstairs now. If you want to talk to Dr. Donahue, there'll be a reception upstairs. Let's thank him very much.
Thank you very much.